Hi, Mike Dolinski here with Taurus. I want to take a little while to talk to you about germination and seedling emergence. Well, the first thing we got to look at is going back and see what happened on the road to senescence. Well, during the mother plant's life, the plant prepared the seed for a new life. That's the objective of sort of all creatures on this earth, to carry on with their progeny. She grew up from another seed and then spent all their life photosynthesizing, taking nutrients from the soil, water, and the sunshine, producing all kinds of proteins, sugars from photosynthesis, and eventually produce seed, which is what we as farmers want to have. First thing is, what's the purpose of the seed? Well, the seed carries all the genetics. It's got all the machinery, the codes to the machinery to make everything work. And more importantly, perhaps, the nutrients to support the seedling till it can photosynthesize and secure nutrients from its environment. So we got to get the seed to fix itself after being dormant, produce a radical and get it up until the plant can photosynthesize and produce its own energy through photosynthesis and grow roots and take up nutrients. So let's take a look at some things that might be important. What happens in the production of the seed? Well, first of all, we get reproduction and we get pollination. And once pollination happens, the plant has a hormonal change. And now the sink becomes the seed instead of the roots and the leaves, which were the sinks during vegetative growth. There's a lot of variability. For example, even in this one spikelet, you can see that these three seeds, and these are the sort of parallel later on in life, are about the same size, and this one's smaller. So right off the bat, just the position of the seed changes its nutrient quality, because it's my belief that these middle seeds here, where pollination first takes place, have the best seeds, because that's where the plant or the seeds have access to the most nutrient available from the plant. By the time we're getting to that stage, the plant is pulling all the nutrients from its basically leaves, shoots, and roots to fill it. But a little further along than this sort of watery ripe or early stage of seed development, a little later, we get the plant filling in all this endosperm. This is the germ here, or the embryo. And you can see that this right here is the coleoptile already forming with the first true leaf already present. So even while the seed is green, it has formed all the genetic material that's hiding behind this little cover here. And when the radical pops out through that cover, you know that you've finished germination. We also know we have a lot of variability based on the topography. Never mind what the plant can get, but what is in the ground. For example, we have variation horizontally and vertically. We know nutrients are stratified badly, especially in zero-till situation in top 3, 4, 5 inches. So as the roots go down, there's less. As you move horizontally over the hilltops into the valleys, you get variances in terms of nutrient availability, compaction, distribution of nutrients, deficiencies of nutrients in certain locations. I mean, sulfur deficiency can be just in big pockets in there that causes tremendous problems for the plant. We get variances in pH, for example, which can interfere with the availability of nutrients. But even after they're sort of just about ready to go into dormancy, we can have problems in terms of final condition of the seed, because here we got piebald, which is a balancing of nutrient problem. Here we got frost, didn't quite make it to the end before it got froze. Here we've got some kind of wrinkling going on. I'm not sure what happened there. We can have heat stress that's going to affect seed. And here we got disease. We got fusarium likely in here that is affecting the viability of the seed. Even here with canola, you can see there's variability. This is just bin run stuff. You can see that there's some damaged material here, and I'm not sure whether this was in the swath or was straight combined, but we got a lot of immature seeds. We want to get all those big purple, black kind of seeds, and that's generally what you get, and they vary in sizes now. In fact, companies are sizing them. The argument about big seed versus small seed, I don't even want to get into. So this can cause all kinds of variability even late in the growing season. So what farmers do is they send in their seed for a germination and vigor test, and that tells you, how many seeds are going to germinate, and you can't change that a whole lot. And then how much vigor are those seedlings going to have? And maybe we can have some influence there by feeding that new shoot and that emerging seedling. So that plant and consequently the seeds produced from those plants that are living in that environment have varying degrees of variability themselves. And the question becomes, can we do anything about it? Well, that's something we'll talk about a little later. So what happens in germination? And germination goes about in phase 1, phase 2. There's typically phase 3, but phase 3 is post-germination, and that's what we call seedling growth at that point in time. So the first thing that happens, and you can see it's a growth curve, goes like this, then it flattens out, and then it takes off again. So during this phase, the plant absorbs water, and a whole bunch of reactions take place. Here, things are being fixed up, and at this point in time, if the radicle elongates and pops out of the seed coat, then we know that it has germinated. So that's the end of germination. If it has not, then it's dead or dormant. Now, germination does not all happen at the same time among all seeds. There is a variance for sure, just because of the position of the seed in the soil, how much moisture is available, whether it's in a saline situation or salt situation versus not, the depth, the temperature, a whole bunch of things affects that. But generally, you'll have the germination in a period of 3 to 5 days. You'll have most of them up and going. So now let's take a little closer look. And I'll use wheat seeds, and then I'll talk a little bit about canola towards the end, and then whether there's anything worthwhile doing about helping out these seeds. In a wheat seed, the only thing alive is this part I like to call the germ, which consists of the embryo and the scutellum. They're live. And this little line along here of cells called the aleuron. The rest of this endosperm is storage material consisting of some hormones in there, but mostly starch. It's basically starch and proteins. And the starch are in granules of various sizes. And when the plant is storing starch, it stores it as either amylose or amylopectins, and then it breaks them down later, and it mixes the proteins and the minerals into those starch granules so that the plant can eventually feed all of this endosperm to support the growth of the new seedling. But that's going to come about in the second part. Right off the bat, when this the seed imbibes water, it causes a big uptick in respiration, and we get a shift in the hormones here. Abscisic acid, which puts the seed into dormancy in the fall, has to be broken down, and that's what happens. And gibberellins, which support germination, are created by the embryo and the scutellum. The gibberellins permeate through the endosperm to the aleuron, which is alive and contains a lot of activity and ATP in there, and the genetic material. So we get messenger RNA making alpha amylase and proteases. The amylases break down that amylose and amylopectin starches here to glucose, and then in the scutellum to sugar. The proteases break down the amino acids that are in here and are transferred out of the seed as either glutamine or asparagine, the two amino acids that are used to transport nitrogen around the plant for the formation of new proteins. Amino acids are linked together and they form a protein. So that's basically what happens. Same thing with barley. Scutellum, aleuron, proteins, starches to glucose to sucrose, glutamine, GLN, asparagine are the amino acids sent up into the shoot, sent into the root, and the nutrients and so on are split somewhat equally, some going more to the shoot than the root, but it's split to support the growth. More detail, when the water comes in, we get a big uptick in respiration and protein synthesis. Now, some of the mitochondria in cereals are alive. They survive that dry down, but the cells are damaged. So they're repaired and some new ones are synthesized. We also get a translation and degradation of stored messenger RNAs. So in other words, some of the stored messenger RNA works, but some of it doesn't. So it's got to be cleaned up. So we get transcription and translation of new messenger RNA. In other words, we get new messenger RNA to produce proteins for the new seedling. We get hormone biosynthesis and deactivation. First, we get deactivation of abscisic acid, and we get biosynthesis of gibberellins. Other hormones are involved in there, brisanosteroids, ethylene. They're all involved in this process, but it's primarily gibberellins. As this period of repair goes on, we eventually get the endosperm weakening and the testa rupture, or otherwise the seed coat rupture, and out comes the radicle. Once the radical becomes visible, germination is over. We now move into post-germination, where we get reserve mobilization and seedling growth. So that's sort of the process. 3 stages. These are just kind of fixing everything and getting ready to go. And then boom, when it's ready to go, we remobilize all those nutrients into the new emerging seedling. Along with the sugars and so on, we have all these nutrients, and this is just some of them. I found this paper, and that's all they recorded, where somebody broke the embryo, endosperm, and seed coat apart and looked at nutrients in them. And there are some key things here. The phosphorus is key for ATP production, DNA production, plasma membrane production. So it's really key. Zinc is involved in all the enzyme groups, and probably zinc and manganese are the 2 micronutrients that are key to actually good seedling vigor. Boron is also in high level because all the cells have to be stuck together, and boron is involved in sort of the structure of the cell walls. You can see that there's, because the embryo is only like 2% of the of the total biomass, but it's a high concentration of some key nutrients in there. When we get to the endosperm, which is like 62% of it, we have all of these nutrients. So you can see there's a lot more copper here than there is there. There's a lot more, pretty well everything in here in the endosperm, because that's going to be fed to that growing seedling. Sulfur is key. And the nitrogen isn't measured in this particular study, but it would be high. But sulfur is needed for 2 amino acids, nitrogen for the rest of the amino acids. So virtually all plants will have to supply sulfur in there along with that nitrogen, which isn't recorded at this point in time. The one strange thing is the seed coat is about 1/3 of the total dry matter. And yet very little of this nutrient in the seed coat will be ever used by the plant or sequestered into the endosperm. Some of the potassium will leach in there, but most of the other nutrients, for some reason, this plant, and it seems to be maybe some evolutionary strategy, don't really have an answer for why so much is tied up there. I know that phytases will tie up some of these cations, and maybe that's the issue. They just can't readily get them, but I'm sure the microbes eventually secure those nutrients. And lastly here, I want to just show this picture from Ismail Chakmak. And you can see how the germ here, or the embryo, and the scutellum are really stained red. And the alluron, that's because this is where we saw those high levels of zinc, because zinc is really key to metabolism, stress management. Stress management also involves manganese, copper, potassium, and zinc in a big way. So it's not surprising that those live cells would have high levels of zinc in them. In fact, on a global basis, zinc deficiency is becoming very, very common, more so in sort of the underdeveloped countries, but even elsewhere now zinc is becoming a significant issue. Now this is what it looks like in the plant or the seed. Here we have canola and wheat at 33 hours after I applied water. You can see that the radical's out, the coleoptile's out, so it's germinated. Here's a close-up. Here's the coleoptile that's going to go up. Here are the two seminal roots, and this is the radicle, which is also called the seminal root at this point, and there are 5 total. So it's germinated. Now here's the coleoptile. I cut open the coleoptile. You can see the first true leaf, so it's ready to go. It was ready to go way back in the fall, in fact. Coleoptile grows upwards towards the surface and is closed to protect that first true leaf. The first seminal roots are already got a lot of root hairs on them and are extracting nutrients. So if you put a nutrient package on the seed, whatever doesn't get taken in by diffusion with the water is going to be picked up by the root hairs. Now, things like zinc and manganese and potassium and phosphorus, which are not very mobile, have to be close to the seed. And that's why a seed dressing containing those is of more importance than maybe a little bit of nitrogen, which has got some mobility and can move around fairly readily in the soil. So it's moving upward, pops through the ground, so now it can release the first true leaf. The coleoptile opens, out comes the first true leaf. And you can always tell the first true leaf because it's round. All the other leaves on wheat are pointed. So at this point in time now, the mother plant has succeeded in her goal of having the first true leaf up and her young baby, for lack of a better term, being able to photosynthesize, take in CO2, produce sugars, and out of the sugars from CO2 and the nutrients in the sun and the water and the CO2, the plant, based on its genetics, biosynthesizes everything. That was the message that was carried through the seed to the new young plant. Let's take a quick look at canola. And I put in these shots here just to go along with the diagrams that I found in the text. Here we have the seed. It takes in water. You can see that this was the umbilical cord that was attaching the seed to the seed pod. So water can enter there and gets in there. It starts to take in water. The testa cracks, and we get reduced ABA, abscisic acid, increasing gibberellins, and we get the cotyledon that is now showing you that it's germinated. Now, canola is a little different because it's mostly oil and protein and not so much carbohydrate. But in the process, the oil is converted to sucrose. It's a long, drawn-out process, but it's converted to sucrose so that the seed can actually move the sucrose or the sugars, the carbon, which is key to growing the plant, into that new seedling, and the plant can feed on the oils and the proteins. And the protein is also converted to glutamine and asparagine for mobility to move up into the plant as well. So here we go. Here's germinated, and you can see very quickly the root hairs on canola are just massive. So it can pick up, in fact, it looks like it's already picked up some of this blue off the seed coat. So we put fungicides on the seed. We put insecticides on the seed. Farmers are now starting to put seed nutrient packages on the seed as well, because it makes sense that if you're going to protect it, you might as well feed it, and especially for things like phosphorus that are hard to get a hold of, and zinc that are hard to get a hold of in the soil, in cold soil, because they're virtually immobile in the soil. So it's got to be right there close to the seed. So here we have germination, and you can see that canola is different than wheat, and that the cotyledons are pushed up to the soil surface, leaving nothing behind. So this little tiny root is supporting this cotyledon. It's like a fist moving up through the soil. When it breaks the surface, the cotyledons open up, and eventually they turn green. So now it can photosynthesize, and it'll start producing new leaves. And I just want to show you what's down below here in this little set of cotyledons. I point this out because many farmers don't know where to look when we talk about, a frost that occurs, and take a look and see if the growing point is black. This is the growing point, or the apical meristem, and this is the first true leaf, and this is already the second true leaf. So if you get a late frost and you think the leaves have been, say, frozen, you have to wait 2 or 3 days and then go and find this apical meristem. If it's black, it's basically dead, and you saw that there was no nutrient left behind, so there is no genetic material there, or stem cells basically, to produce the plant. Compared to wheat, which has the growing point below ground, you can freeze off the leaves and it'll still push out another set of leaves if it's not too far along. And things like peas, where the pea seed sits below the ground, if you freeze it off, generally pea will push out another set of leaves and try and grow a plant. So anyhow, that's where you look for it. Now there's a lot of stresses on seedling when it's growing. You got some cases poor seed, you got cold soil, and heat is important. The cooler the soil is, the longer everything takes. So in cold soil, it's maybe more important to have a nutrient seed dressing on there to help that plant. Salinity issues are a problem, where you got salts in there and the seed can't imbibe enough water because the soil is pulling the water actually out of the seed. And you have dry conditions, wet conditions, cold conditions, seeding depth issues, fertilizer salts, variable seed depth, variable seed across that field. So is there anything you can do? This is what it's all about. If you want to try a nutrient seed package, we got hundreds of farmers using it across the West, and once they use it, they sort of don't move away from it very often because it's relatively inexpensive and it balances out any imbalances that are in the seed. But we developed this based on rational thinking. We know we need some nitrogen because plants always need nitrogen. If they're growing, they need it for protein production and growth. We always need lots of phosphate, and that's the key thing here, because especially in cold soil where roots are growing slowly and water doesn't move a whole lot, neither does phosphate. You got to have it right on the seed or right really close to it. Soluble potash for turgor pressure, and also to help that little seedling control stomatal opening and closing. Potassium is also key in terms of turgor pressure and moving and expanding the cells to push the shoot up and push the roots down through the ground soil. Boron to stick the cells together, iron in the electron transport system, manganese for breaking that water molecule, the first thing that has to happen before a plant can photosynthesize, and zinc for all kinds of enzyme activity. Zinc is the only micronutrient that is in all the categories. There are 6 categories of enzymes in plants, and each category has some enzymes with zinc in them. Well, here's a seed that's treated with active prime, and you can see that, and I've noticed this in other things that I played with when I was doing some work way earlier on in my career. The distribution of nutrients doesn't seem to be uniform, because some parts of the seed are kind of smooth and nothing sticks to it. But I always see that there's more that sticks to the germ end of it and to the bush end. Well, the bush end I can understand because it's kind of hairy, so if you get some particles in there, they'll stick. But it's right there where the seed is going to germinate. So as soon as it pops out and the root hairs are getting grown, it can pick it up directly, or maybe took some in when the plant was taking up water in terms of some of the ions were in the water solution. But this gives it doesn't seem like a whole lot, but relative to what's in the soil, it's fairly concentrated for the plant to pick it up and utilize it. This is about the last thing you can do to support that, though, germination. You put on a nutrient package, fungicide, maybe an insecticide, and your plants got the best chance of making a go of it. Thanks a lot. Hope you learned something. Good luck this spring, and always remember, the plant knows what to do.